Okay, well, first, thank you guys so much for the invitation to be here and from the organizers. I'm excited to share some of the work that's going on in my lab, which is focused on our rare disease patient population in Kansas City.
So there's a same.
Uh-oh. So this is my title slide. In focusing a lot of our work on scalable technologies and doing this work in patient-derived cellular models. So to get started, I want to lay the foundation. Some of this may be a middle school or high school refresher, but let's get on the same page. So virtually every cell in our body contains 46 chromosomes. We have 2 copies of 22 autosomes, and then our 2 sex chromosomes. Right? And distributed across these chromosomes are roughly 20,000 protein-coding genes. And the spatiotemporal expression patterns, the when and the where, dictate phenotypes. So in the laboratory, we think a lot about cell models, so things that impact cellular proliferation and things like that. But when we get down to it, cells make up our organs, and organs make up our body. So small changes in our genes can actually impact human phenotypes, right? So that's the ground truth that we're working with. Here's an example of this from our laboratory from very early on that I thought was just amazing. So this is a picture of something called an embryonic body. This is a 3-dimensional kind of ball of pluripotent stem cells. And we form these when we're working with various differentiation models. But when we form these, this is from a healthy or unaffected individual. You can see that it's this nice, tight, round ball. This is what we typically or almost always see when we're making these kind of cell models. But here's one that we generated from a patient with a rare disease. This is a diagnosed patient. We know what disease they have. But even at this very basic level of building these cell models, you can see how a single letter change, and we know this is a causal variant for this particular patient, we can see differences in these cellular models at these early stages. And this tipped us off very early on that these were going to be very, very powerful tools for exploring rare disease. What we're seeing here actually in real time is that underlying genetics can impact disease development, or healthy development or disease. It can also impact disease progression. And then therapeutic vulnerabilities and responses to therapeutics. So this is really important stuff. So now going back to a little bit of history, for as long as that I've been in this game, we've used genetic manipulation for trying to build models of a disease. I kind of swiped this image, but here in this example, it would be taking mouse cells and then engineering in genetic changes into those cells where we're trying to determine if specific genetic changes are actually leading to disease. Right, so we've been doing this for a long time. CRISPR came along and accelerated the process, but really it's kind of been a pretty standard approach. So we've been doing this since the late 80s, but in reality this is actually pretty time consuming to build cellular models these ways. It's expensive to build them well, and one of the large challenges with this approach is that it's a hypothesis-driven design. Right? We do a lot of work to try and predict what types of genetic variation or variants that we see is leading to disease, and then we test that. And more often than not, I think we're wrong, right? And that's just the scientific method. Now, in the early, I guess mid-late 2000s, the process of iPSC reprogramming came along. This is a method where you can take, in theory, any cell from a human body. You can introduce reprogramming factors. These are proteins that will basically reset the genome back into an induced pluripotent state. Now that's really powerful because then you can take those stem cells and you can differentiate them down different developmental trajectories to try and understand disease in that manner. Again, this has been around for a long time, but it turns out it's also really time consuming and even more expensive to do until recently. But the power here is that this is ideal for discovery biology because we don't have to know what genetics are causing disease. We have it there. It's harbored within that cellular model, but now we can probe at it and try and understand what it is, what's causing disease. So these are really great discovery biology models. So that's kind of a little background on setting the stage for what I want to talk about today. I didn't want to come straight out of the blue, but now I'm going to introduce what's actually going on up in Kansas City. And so I came to Kansas City in about 2019, and right about that time, we launched what really is one of the most aggressive rare disease programs in the world. And this program is called Genomic Answers for Kids, and in the program we're aiming to sequence the genomes of roughly 30,000 children impacted by rare genetic conditions. So you can think of this largely as undiagnosed rare disease. And then in this process, we're developing the largest pediatric data repository really that's ever been made. And then my lab has been tasked with building tools and technologies to help us and facilitate our understanding of the genetic variation that we're detecting in that patient population. Many years ago, we had this idea that if we could just take these biospecimens from our patients. Now, getting back to that, when a patient enrolls in our program, they provide a blood draw, and we use that to perform whole genome sequencing. And then we cryopreserve or we freeze down residual blood cells. And we thought, well, it'd be pretty cool if we could take those blood cells and then reprogram them into induced pluripotent stem cells, and then we could have this huge biorepository of cellular models. But we came into this challenge of this is really time-consuming and expensive, too much so so much so for an academic lab. But again, the model was this: we would take these residual blood cells, and then you can go through this reprogramming process, and this is what a colony of induced pluripotent stem cells look like once you've achieved that reprogramming. So we said, okay, let's do this. Let's reach out and find a contract research organization that's going to help us along this journey. And what we quickly found out about 4 years ago, that the cost of this was about $10,000 per patient. When you sign up for it, the turnaround time is about a year till you actually get an iPSC line back in your hands. This is per patient, so you do this one off. Also, a caveat is that in a lot of these CROs, you sign a waiver that says, hey, this is probably not going to work, but you need to pay us anyway. And so that was really a non-starter for us. And so we brought this technology in-house, and through really a Herculean effort by a research scientist in the lab, Dr. John Means, he brought this cost down to about $200 in reagents. We were able to get this accomplished in about 2 to 3 weeks per line. And now also keeping in mind that when you contact a CRO, they want lots and lots of blood from patients to engineer these lines. All we have is cryopreserved residual cells from previous genetic testing. So it's actually more cost effective for us to do this in smaller plastics, so in 12-well dishes. So again, it's more cost effective to do 12 lines at a time. So this is a figure from a recent paper where we went on like a 6-month campaign of just reprogramming and engineering these models. We did about 300 for that project. This is for various purposes, to build models for studying diseases, for evaluating therapies, and also for diagnoses. So these all had purpose. They weren't just to do them. And to date, that number's, I think, well over 400 lines that we've generated and have in-house. One of the things that I was really concerned about early on was that there would be some intrinsic features of disease, of certain rare diseases, that would preclude us from using this approach. But really what we saw, that as long as we had 300,000 PBMCs in hand, we were successful at generating these models. So it was more a function of how much our starting material we had than anything intrinsic to disease. We've been able to get these, in some cases, down to about 5,000 cells. We've been able to still reprogram those lines. So this is a really a robust approach. I guess it's worth noting, so typically when we do a blood draw and isolate PBMCs, we get about 1 million per mil. So 300,000 is less than half a mil of blood that we're using. We were tasked with doing extensive validation on these iPSC lines, and here's just some immunofluorescence. These are different proteins that are markers for pluripotency, and everything we checked, you could see these are all expressed in these iPSC lines. We also did extensive karyotyping on these. So when you do this process, it's not unheard of to end up with genomic instability, where you can either end up with aneuploidy, or even cases where you have a trisomy, where that third chromosome is dropped out, and they're a programming process, so you're not really developing models of disease. But what we've seen through profiling many iPSC lines is that we have really robust lines. And then we've also gone through, and you can do, this is called the trilineage differentiation assay. So you take these stem cells and you differentiate them down to ectoderm, endoderm, and mesoderm, the 3 different germ layers, as kind of proof that these truly are pluripotent. We can make any of the germ layer, or at least lineages from any germ layer, out of them. Great. And so that sets the stage. So we have this really robust pipeline for generating induced pluripotent stem cells. And now what I want to go into is what we're actually doing with these things. So it was a lot of work to get to be able to make these, but what's just as important is what you do with them. So in my lab, all of our work is kind of right now occurring in the cardiac space and in the neuronal space. And really, that's driven by our patient population and what we see the types of clinical indications. I think 80% of our rare disease patient cohort has neurodevelopmental challenges, right? So this is a really relevant cell model. The other reason, I guess we'll move on to the next slide. The other thing that we're doing is all of our work is done in organoids. So organoids are three-dimensional cellular models that recapitulate organ development and function. They're not actually organs, right, for like transplantation or anything, but they're just little three-dimensional models. And here you can see a video of a cardiac organoid we generated from unaffected individuals. One of the reasons we started with cardiac is that we knew that if you do this properly, they sit in a dish and they contract. And so you can actually see what you made and validate that you were, in fact, generating cardiac organoids. Now, if you do the same experiment and you engineer or generate a cardiac organoid from a patient with a muscular dystrophy, that's what it looks like. So this is actually a video, and you might kind of see this move slightly. And in muscular dystrophies, a lot of times we think of skeletal muscle, right? Because that's the outward presentation, but our heart is actually really one of the most important muscles in our body. And so here you see, this is disease in a dish. This cardiac organoid is not contracting. So this was also early on we saw that we could observe clinical phenotypes, or at least some proxy for clinical phenotypes in a dish. So moving forward, I want to kind of talk about the two different arenas that we're doing our work in. And one is the precision and personalized medicine space, which is highly relevant to this crowd, I think. But I also want to touch a little bit on the end of kind of the hypothesis-driven exploratory research we're doing with these models as well. So we're kind of diversified in what we're trying to accomplish with these things. But we're going to start with precision and personalized medicine. That's where we've had kind of the most activity to date. So again, working on some terms, precision medicine, I guess you guys are probably familiar with it, being a precision medicine venue. But this is kind of the idea of precision medicine shortly after the Human Genome Project. The idea that we were going to get genome sequenced, and that was going to tell us everything about disease and what drugs were going to work. And so we had this idea, we, not me, but people that preceded me, in precision medicine. And the idea is that if you sequence, in this example from this paper, tumor DNA, and this is an approach that's been very successful in certain oncology spaces, you could understand a tumor and then you could predict what types of therapeutics are going to work very well for that particular class of disease or cancer. So this has been successful, as I mentioned, in oncology, but when you get into genetic conditions or congenital conditions, this actually doesn't work very well at all. And that's not that it's not a good approach, it's just not appropriate for that. But in recent years, this has been stratified kind of into an area that we call genomic precision medicine, right? Using genomic information to predict therapeutic responses. More recently, we're doing a lot of work in functional precision medicine. So the goal is the same, but the way we get there is different. In functional precision medicine, this is done largely through ex vivo testing. So in cancer, for example, if we can get our hands on those cancer cells, then we can actually do experiments in a dish and build an empirical evidence base to say, hey, this is what actually worked for that actual condition. And this has been really, really effective in oncology. Again, this until recently really hasn't touched the spaces of genetic or congenital defects. Okay, so this is something that we wanted to set out to do, right? I told you we built this platform for generating these cellular models of genetic disease. And so really early on, the idea was to be able to take a biospecimen from a patient, to build cellular models and do experimentations in a dish. But the big thing we wanted to accomplish was, this is important in and of itself. It's very important, right? And we can understand disease, and then we can think about children in the future that may be impacted by that disease and what we can learn. But our goal, we're really laser focused on doing these experiments in a timely manner that can actually inform the clinical care for the patient from which that biospecimen came from. And that's what I'm going to talk about. So again, getting back into parsing through these terms from precision versus personalized medicine. In these approaches, we use genomic data and biospecimens from our patients, so the input is the same, right? The goal is the same as well, right? We're trying to identify a therapeutic approach for an individual. And the kind of data that we're interpreting is also largely the same, genomic data and experimental data. The difference here is that precision medicine, we're focusing on existing approved medications, things that are available right now, which one's going to work for this particular indication, right? Whereas personalized is we're talking about tailor-designed, N-of-1 therapeutics, and that therapeutic approach is almost, likely only going to be efficacious for that one individual, right? So that's what distinguishes. Who's the target crowd we're pointing at? So I'm going to talk about both of these areas for what we're doing, but I am going to start with our personalized medicine because that's where we've had, again, the most success thus far. This is a plot I showed you earlier, right? This is the kind of data points of the number of cell lines that we've generated, but one of the things that's easy to get lost in is that each one of these data points is actually a child, right? That point right there is a child with Duchenne muscular dystrophy, okay? We know that this is a confirmed causal structural variant within this child that causes disease, right? So this is something we wanted to go after, and I'll explain why in the following slides. Here, again, you saw these earlier, those cardiac organoid images that I showed you were actually from this patient right here. So this is cardiac organoid generated from this patient right here. Okay, so the nature of this patient's disease, or the underlying genetics, this patient has a structural deletion. So Duchenne muscular dystrophy is caused by mutations in the dystrophin gene. This particular patient has a structural deletion of exons 46 through 53. And so what happens in that patient's cells, and also this is an X-linked disease, so it affects, more prevalently, it affects boys. And so this is a male, one copy, has a structural deletion, right? And so what happens in the patient, that patient's cells is that the gene is made, but due to this splicing of exon 45 to 54, the gene is out of frame. The transcript is out of frame, so no productive protein is made, right? And that's what causes disease. We know that. The reason this patient stuck out to me in particular is in 2021, the FDA approved a particular type of therapeutic for just such a structural deletion for the treatment of Duchenne muscular dystrophy. So this treatment is called an antisense oligonucleotide. These are short synthetic nucleic acids that you can put into cells and alter gene expression in various ways. So you can knock down gene expression, or in this case, you can cause exon skipping. So you prevent exon 45 from being included in the transcript. So now exon 44 will connect to 54, right? And so you'll never gain back that genetic information. But what you do is you knock that transcript back in frame. So you do make a protein, and that protein has been shown to be therapeutically beneficial, right? So here we have a FDA-approved therapy that this patient could be treated with. So we had the question of what happens if we generate a cell model from that patient? Can we recapitulate that therapeutic response in a dish? Okay? So here's an example of immunofluorescence expression data. Again, this is a cardiac organoid from an unaffected individual. This is a cardiac marker, and this in green is dystrophin expression, and you can see in the patient that expression is completely absent. When we treat with the ASO and introduce it back into the therapeutic into the cells, you can see that green coming back. So we're seeing dystrophin being knocked in frame, and this is actually what happens in patient cells. Now with these cell models, we can actually go a bit further and do functional data. So here's a video of a cardiac organoid contracting. So you can see it contracting. And the reason this is green, so this is a cardiac organoid that's been stained with a calcium indicator dye. So that green flash you're seeing is calcium release, and that's associated. That's a part of the contraction process. So you can see the wave of calcium flux during contraction. When you generate this from a DMD patient, you see it just kind of sits there. There'll occasionally be a calcium flux, but it's decoupled from contraction, so you can see the functional breakdown again in a dish. But when you treat with the ASO, the therapeutic, you recouple that calcium flux with contraction, and this thing starts beating again. So again, you're actually able to see this therapeutic response in a dish. So that's really exciting, and it sets the stage for where we can go, but maybe it's not as exciting to say, hey, this FDA-approved drug actually does what it's supposed to do. So we went a step further and went back into our patient cohort, and it turns out we have another patient, this one right here. This is a patient who also has Duchenne muscular dystrophy, but their mechanism of disease is quite different. For this patient, they actually have a deep intronic variant within the intron between exons 43 and 44. And that variant causes a novel splice acceptor site and incorporation of a cryptic exon and then premature transcript termination. So again, no dystrophin proteins being made in this patient. But actually, this turns out to be a really great target for an ASO therapeutic. If we can just target that single nucleotide variant with an ASO, then we can maybe generate an individualized therapeutic for just this patient. So that's exactly what we did. Here you can see the pre-messenger RNA transcript for the dystrophin transcript. You see this is the intron, this is the variant, and this is that cryptic exon. And we developed an ASO that really straddles right over that variant site. And when you do these experiments, you see the exact same result, right? So here you're seeing restoration of dystrophin expression, and you're seeing that restoration of contraction. So really, we've developed a method where we can rapidly screen through personalized therapeutics. Now, one of our motivations to do this was a 2019 study from Tim Yu's group at Boston Children's, really like an amazing study where they took a patient with a condition called Batten syndrome. That patient had a retrotransposon insertion in the causal gene, so they had a splice disruption, really the same mechanism we just talked about for DMD. This Batten syndrome is associated, or Batten disease is associated with seizures, right? So what they did there was they generated an ASO to prevent the splicing disruptions, right? They administered this intrathecally to the patient, and what they saw is a significant decrease in the frequency and duration of seizures. So they really set the stage. This is absolutely doable, and it absolutely can be used to treat a patient. The challenge here is it takes a lot of work and a lot of heavy lifting, and so getting from A to B as a process that took like 16 to 18 months to get through. And now we're not treating patients right now, but what we've really done is shrink down that preclinical timeline so that for us to get PBMCs, sorry, it's a little pixelated, and generate these iPSCs and organoids, right, in this time, the 2 to 3 weeks, we can design ASOs, and generate these organoids and then profile the ASOs in the mature models. We can do all this in about 6 weeks for cardiac organoids and about 8 weeks in the neuronal space. So it really, really shrunk down the time frame to do that and the scalability that we're able to do that at. So there's a lot of challenges that I won't talk about now with actually moving forward to treating patients, both on the financial and regulatory side. But in doing this work, we actually blew right past what really is the most practical application for the technology, which is going back to precision medicine, right? Rather than trying to develop new drugs, looking at drugs that are already available, seeing if we can explore that. Okay, so for this, I'm making a shift into brain organoids. I did a lot of talking about cardiac organoids, but now we're going to shift into the brain. This is a picture of a brain organoid, right? So these are not sentient brains that are just sitting in the lab. They're about the size of a letter on a penny. We call them brain organoids. Really what they are is kind of a heterogeneous ball of neuronal cell types, right? And so we're looking at really fundamental basic phenotypes using these models. And again, we've done extensive profiling. There's lots of different flavors of brain organoids that you can choose to make. These are cerebral organoids. They're kind of a mishmash of a lot of things in the brain. And we've profiled those and validated these. And so one of the things we wanted to see, like I just talked about, was can we recapitulate clinical phenotypes in a dish? And so if we turn back to the patient population, what we see is like 70 to 75%, I think, of these patients in the rare disease cohort are impacted by seizures, right? So we were interested, can we profile neuronal activity in a dish? And so what we've done is we've cultured these three-dimensional brain organoids, and then we can actually soak them in the same calcium indicator dye, right? So it's going to glow green. And then you'll see occasionally a blip here and here. It's pretty, don't blink, you'll miss it. There it was. So that's the regular amount of neuronal activity we see in a 4-week-old brain organoid. Now, when we generate one of these from a patient with seizures, it looks like this, right? It's really a qualitative observation. It's night and day. We've done this for more than 36 different patients with diverse causes of disease or mechanisms of disease, and many with undiagnosed disease, so we don't know why they're having these. So what this is I think really just a basic lack of inhibition for neuronal activity, so we can actually visualize these seizures in a dish. To do this, right, we have to have a pretty nice microscope, and we have to have cameras and things to do all this and process the data. It takes a lot of work. But really, as we're all sitting here looking at this, all this is light. So we have adapted this to a microplate reader, right? Just a machine that can read light. And so we generate these organoids in 96 well plates, so 1 organoid per well, right? And then we can just set them on this microplate reader, and then instead of spending hours and hours processing videos, just within a couple of minutes, we can actually profile neuronal activity, and it looks like this. So here's 4 different individual organoids from an unaffected individual. You'll see the occasional blip. And then here's 2 different patients that are impacted by seizures. And again, you can really see that quantitative activity. But it's really night and day. Okay, so this, moving into the next step, we wanted to see kind of in a pilot experiment, what happens if we treat these with anti-epileptics, right? So that's what we're getting at. Can we predict drug responses? So here I'm showing you, this gets a little busy, but it's kind of building on the data you just saw. So each panel here is a different condition, and in it is 8 different brain organoids, so there's lots of redundancy repetition in here. So all these different colors just represent different individual organoids. So here's from an unaffected individual, those occasional blips, and then here is 3 different patients, and you're seeing those seizure phenotypes. Okay, so here what we've done in this pilot experiment is treat these with just a selection of 3 different anti-epileptics to see what happens. And so this is an interesting area for seizures, this clinical indications, right? There are actually symptomatic treatments, anti-epileptics, but they show a very high degree of patient-specific responses. So there's more than 30 of these things FDA approved. Only 1 or small subset is going to work for a given patient, and really you have no way to know what it is. So standard of care is dose a patient, wait 3 months, see what happens, increase, decrease, change. There's kids in our program that have been on this odyssey for more than 10 years of trying to find an anti-epileptic. So this is a serious challenge. So in a pilot experiment, what we did is we just treated with these 3 different anti-epileptics. I just labeled them A, B, and C. And here you can see for patient 1, A worked, C worked, B didn't. And then looking down, you do see we were able to recapitulate patient-specific responses. Just in a pilot, we can see that. And what's worth noting is drugs A and B, I don't actually remember the exact names of them, but they work through the same mechanism of action. So on paper, you would anticipate that these 2 drugs would behave the same way in all patients, which they did here, but not in these patients. And it highlights the need for these kinds of experiments. So after showing that we could see this patient-specific response, we were able to secure funding to go into a retrospective study, again, leveraging this rare disease patient population. We can go back to electronic medical records, so we can find patients that have seizures, we can see what they've been treated with, and we can determine if we can recapitulate that in a dish. I'm only going to show a snapshot of this, but here's one patient as an example, undiagnosed rare disease. This patient's been treated with a lot of different therapeutics, and here we can see what happened. I'm going to highlight 3, and this is for 2 reasons. There's a practical reason. These are all, you can get these off the shelf. They're not controlled substances, so they're pretty easy to get in the laboratory. But also we saw a variety of responses. This first drug had side effects, but it did not have any impact on seizures. The second drug actually increased seizure activity, right? So that unfortunately happens sometimes with these classes of drugs. Sorry, and then the third, you can see that the child's been seizure-free for 6+ years. So we generated brain organoids from that patient. Again, we never had to go back to the patient. We could go back to cryopreserved blood cells, make the brain organoids, treat with the drug. This is a dose response. So in each row here, you're seeing an increased response for the 3 different drugs. That's a little busy, but we can kind of reduce that down to like an area under the curve type of analysis and make it a little more digestible. That first drug, you can see no major change. The second drug, increased activity, and the third drug decrease. And so we've gone through about, I think about a half dozen patients where we've been able to recapitulate clinical responses in a dish, right? So we feel really, really good about this. So moving forward, we're currently kind of just past the stage of doing what would be considered, I'm learning the language around the clinical stuff, this would be considered a case study. This is an email I got, it's hard to believe, almost a year ago, but from June last year from a clinician, from a patient having seizures, from a suspected gain of function variant in a calcium channel. They were interested in this for 2 different reasons, for a potential ASO therapeutic. So this is something you could target. If what I'm telling you that we could do in the lab is actually real, we should be able to get this done in about 8 weeks. So that's how much time it would take for us to get a blood draw, generate stem cells, make brain organoids, and treat with a patient-specific ASO. So here's the actual empirical data. This is 8 weeks and 1 day later, where you see this increased neuronal activity. The plotting kind of changes as we've gotten better about analyzing this data or understanding it better. But when we treat with the ASO, you see this dramatic dip in activity. So that was really cool. It validated the pathogenicity of the variant. Again, this is not being administered to a patient, but it's a preclinical lead. But what we were able to do in the exact same dish that we did all these experiments was profile calcium channel blockers. These are 3 different drugs that the clinician was considering using for the patient. And in the same dish, we were able to profile all 3 of these. Again, this is a little busy, but here's a plot of neuronal activity. And so we saw that nifedipine was the only drug that was working well for this patient. So again, this kind of reinforces that this is a calcium channelopathy, but what the clinician actually went ahead and dosed the patient with nifedipine, which resolves seizures for the patient. So now we're actually at a stage where we're going through kind of at a single patient level and starting to inform clinical care. And so seizure is the indication we're doing now. So this impacts about 470,000 children in the United States. We also have collaborations focusing on ADHD, another disorder that has a large degree of patient-specific responses, and we actually do see neuronal phenotypes and brain organoids derived from those patients. We're exploring depression, although it's too preliminary to see what we can do with that, but we do have more advanced studies in cardiac arrhythmias, again, another area that has therapeutics with high degree of patient-specific responses. And one of the really exciting things in the lab is that we're migrating away from these plate reader assays and moving into these microelectrode array systems. And so these are basically 96-well dishes, but instead of using calcium flux as a proxy for firing, you actually get high-dimensional electrical data right off the individual organoids. And I think with this, we'll actually be able to go deeper into studying seizure phenotypes and understanding, again, and deeper level of what electrical activity looks like in these models. Okay. So I think I have a little bit of time left, so I wanted to break over into what we're doing kind of more on the research side of things. Like we're doing this kind of technology advancement, but we also have questions. We want to leverage our rare disease patient population and our access to these patients to advance our understanding of disease. So one of the projects that we have is focused on the systematic identification of rare genetic variants that lead to congenital birth defects, and this funding is focused specifically on brain abnormalities. So one of the things that we're able to do, we can take these stem cells from patients with brain abnormalities, generate brain organoids, and try and understand what's unique about them. And so here's an example, again, just a bright field image with replicates of brain organoids we've generated from a healthy individual. But what we see, these are 6 different patients that have structural brain abnormalities. But even at the level of a brain organoid, we see that these have distinct phenotypes, structural phenotypes in the actual dish. All right, so we're actually able to explore these. And so we have a few different programs going on. Coming up in the next year or 2, we're moving into structural transcriptomics, where we're basically going to be taking these organoids, right, that you can see look different. We'll be sectioning them and then be performing this spatial transcriptomics to understand what's expressed where and what's unique and how these diseases potentially develop. So that looks like something like this. This is I would say it's a newer technology, but it's been around now for maybe half a decade because time flies. But you basically layer these onto a bar-coded puck, which then you can dissociate, and then you can form near single-cell sequencing, and then layer that back onto the image of the actual of slice, so you can see kind of spatially what was where. Okay, and then some of our other funding is focused on, again, neuronal phenotype, specifically with seizures. And so here's an example of 6 different patients. I mentioned the seizure phenotype. We see this uniformly across all of our patients with seizures. And so leading into this project, we knew we could profile seizures, we could see changes in seizures, and we also knew kind of from the early talk on personalized therapeutics that we had a platform for identifying or screening through personalized therapeutics. So we have an idea for a platform that looks like this, where we could potentially take a patient, sequence their genome, right? We could explore their genetic variation and profile that. Now, jumping out here for a second, the variants that we talked about earlier, particularly the personalized therapeutics, are focused on targeting intronic variants. Introns make up 35% of the human genome, right? So it's a lot of space. Protein-coding genes make up less than 2%. So introns are 35% of the genome, and we really have no way of understanding. There's no good tools for understanding what goes on in there or what impacts RNA splicing, and so it's really just it's just a space we can't explore. But if we could identify intronic variants that disrupt splicing and identify those, we could generate brain organoid models, we could develop personalized therapeutics, and potentially, this is in very light gray, maybe one day actually be administering to these patients. Now, on the bottom here, we have fish. So we do have cases where we identify intronic variation in genes where we don't know what they do. We can rapidly model those in zebrafish. So you can do this in about a week. And we have a collaborator at the Stowers Institute in Kansas City who's doing the zebrafish component of this. So how do we identify? So I'm telling you here, prioritizing intronic variants. How do we get to that space? How do we identify intronic variants that cause splice disruptions? So we built a tool for this. So this is a GFP mini gene. So it's a gene on a plasmid that we can just introduce into cells. And when you introduce into cells, the cells will glow green, right? So in this GFP, we cloned in a human intron, a short little intron. So now splicing occurs for this mini gene. And then right in the middle, we put a cloning site. And on that cloning site, we can drop really anything we want. For example, a 100 nucleotide sequence of interest. We could look through all of our patients in our rare disease program, identify intronic variants we think might be consequential, and then just design oligos, about 100 nucleotides, drop it in and see what happens. And so if we put these in cells and they don't impact splicing, this blue sequence, then we'll get a PCR product. That's just a clean exon-exon junction, nothing to see here. If we have a variant that disrupts splicing, you'll get a PCR product that's going to have a lot of stuff in the middle, right? And so this is basically a binary, did it splice right or did it not splice right? And we can go through one sequence at a time and test them. Now, that's not tractable with all the variation that we're detecting, so we've adapted this to a high throughput format, where instead of looking at one sequence at a time, we look at hundreds of thousands of sequences at a time. Right? And we clone large libraries of sequences into this little spot right here. So we have a library of plasmids we can put in cells, and then they'll all be expressed and various splicing outcomes will occur. And then we've developed a multiplex barcoding system where we can just toss these on a NextGen sequencer, right, and get splicing outcomes and determine which variants are impacting splicing. It's a little technical, but I promise it works. And so here's a very rudimentary plot showing splicing outcomes of an early experiment where we profiled 4,600 intronic variants. And we're able to identify up here in these red dots. These are individual variants that we identified in patients that in our reporter system led to novel splice acceptor sites or novel splice donor sites, right? So we're able to identify these things. Now going back to the data, something that I was not expecting but was pleasantly surprised to see. Here's 3 examples of variants that we spiked into the library because they were reported in the literature and we were known to cause disease. And what you see here in red is the variant, and then you see in blue the cryptic exons. So we know that this is what happens in the patients, and what we're seeing is our ability to recapitulate at single nucleotide resolution the outcomes that we were seeing in the patients. Right? If you're a splicing guru, which I am not, but when you get into the canonical splicing sequences on the intronic side, there's always, typically, canonically, always an AG, and you can see how those variants actually activate that AG. But what's also cool is we can go through the data and identify different types of variants. For example, here, again, green is GFP, the gray is the intron, blue is the cryptic exon that gets incorporated. You can identify variants that occur within the exon or within the intron. These are splicing enhancer sequences that activate splicing events upstream or downstream. This is really valuable for us as we think about moving forward with designing personalized therapeutics and understanding, do you design a therapeutic targeting the variant or where the actual splicing event occurs? So that's something that we'll be exploring in the future. One of the things I want to make sure we touch on is that there are tools out there, computational tools, for predicting splicing outcomes, right? So you can just plug in sequences into, machine learning algorithms, and it's going to spit out what's actually going to happen. So we tested our data from the empirical experiments versus the computationally predicted data. If these overlapped, you would expect these to be all bunched up on the axis, but what you see is virtually no overlap between the 2 approaches. And that's not to say that one's right or wrong or better or worse. Maybe they just detect very different things. Now we're always going to hedge our bets on things that we actually see in the dish. So here's an example. Again, these are all data points, but every data point is from an actual patient in our cohort. So this data point comes from this particular patient here. This is a deep intronic variant within the ANKRD11 gene. And what sticks out about this patient to us is that they have seizures, so they have a functional phenotype that we know we can profile. And they also have this structural brain phenotype, so things that we know we can also model structurally, right? So we can go back to this patient. The idea here is that the patient has this disease gene that I've just told you about, and that perhaps this is what's happening, this splice disrupting event that prevents splicing and no protein being made. So here's a brain organoid we generated from that patient, and this is the immunostaining for this gene, ANKRD11. You can see it expressed in a healthy or unaffected individual, and you can see low levels of expression in the actual patient. Okay. Again, we look at the structural phenotype. We can see that this patient actually does have a structural phenotype with their brain organoids. And then here's the functional phenotype. So here's a healthy individual, again, an occasional blinking, and then here you see kind of the seizure phenotype. So we're able to do all of these things, and that's quantified here, right? So we're able, again, to recapitulate this patient's disease phenotypes in a dish. Also, we wanted to validate that this was a disease, a causal gene, so we've knocked it down in zebrafish at our collaborators over at the Stowers Institute. And what you can see are these defects in brain development, specifically a reduced brain size. And then also, this is really cool, this touch response, where if you touch a wild type fish, it'll kind of move around. But this proxy for seizures is this erratic behavior in response to the touch response. So here you're actually seeing when you knock down that gene in a fish, it exhibits the same phenotypes as disease. Great. So the idea, if this all adds up, is that we should be able to use an ASO to reverse the phenotypes in this patient. And so here's the ASO we designed. This is tough because in the DNA there was a poly-T track right upstream of this, so it was kind of hard to get anything specific. But we got it right over here, right over the variant in that cryptic exon. And here you can see we get this restoration of protein expression and also see a resolution in the seizures. So again, we have a preclinical tool that's been able to validate the pathogenicity of the variant and also serve as a preclinical lead. So these are the kind of approaches we're taking. And we have a lot of collaborations going on up in Kansas City. The rare disease work I've talked about is largely associated with diagnosis, is done with the Genomic Answers for Kids program. We have our ADHD programs, so that's focused on patient-specific drug responses. We have programs focused on response to infection. It turns out that there's some common viruses that a small subset of children will develop very severe neuronal complications. And so understanding, using cell models derived from those patients, what may be leading to those. And then early stages of studying oncogenic transformation, specifically in brain cancers. Great. So this is kind of the overview of what I've told you about, and kind of our mission is to build a platform where we can take blood draws or biospecimens from patients, engineer cell models, and learn things, and again, in a timely manner that can actually influence clinical care. And we're doing a lot of collaborations. We're interested in collaborations. But the way this works typically is you have an investigator-initiated research project, that would be some of you, and then you want to develop a patient-derived model for the disease of interest. But what we ran into is this huge bottleneck of just our labs just doing all of these collaborations. So to make this actually scalable and work efficiently, we built out a patient-derived modeling platform where we can actually just take blood draws from investigators and return these stem cell models. So here's an example for 10 mils of blood. That's actually a lot. We can do it with a lot less of blood. We can actually isolate PBMCs, engineer these cell models, and return cryopreserved iPSC lines to individual investigators. We're kind of not exactly like, I'm not pitching this and selling it. We're thinking of as a cost recovery service that it would be somewhere around $1,000 per line. If you were interested in doing, you could send this blood and get a model back, and then you can do whatever you want to do with it. Also, we're exploring ways in which we can engineer the organoid models at scale and distribute those back to investigators as well. That's a little bit more expensive. Okay, and then I think I'm good on time. I have one last thing I wanted to cover, which I think is really important. It's a cool study that we're getting ready to write up and send out soon. But this stems from ASO-related work. On a disease called Friedreich's ataxia. This is a rare disease caused by repeat expansions in the intron of the frataxin gene. And the end result of these extreme extended repeats is you get a decrease in protein expression. It destabilizes the protein. So work had been done. Actually, this is David Corey, who's my graduate school advisor. This is work that was done after I left the lab, but I'm familiar with it, very familiar with it, where they targeted that repeat expansion with various oligonucleotide approaches. and they were able to show that you could restore frataxin protein expression and commercially available cell models of disease. So the idea was that you would have this ASO therapeutic to treat children or individuals with Friedreich's ataxia. So data looked really good on this, so they went into mouse models, and this failed miserably. I don't say miserably. I mean, it didn't have the effect that it was intended to have. And so they wrote up a story about, or a paper about why maybe that was. So what's important to keep in mind is that mice do not get this disease. We have engineered models to study this disease, in mice, but it's not a disease that the mice naturally get. And why that's important is because the FDA just now, a couple years ago, dropped the requirement for animal testing prior to moving into clinical trials. So what we were thinking about, what if that mouse work had never been done? And what if they had just gone directly into patient-derived organoids? So we happen to have quite a few Friedreich's ataxia patients in our program. Here's an example of one of them. And then here is this idea, this repeat expansion of the frataxin gene, and here's the ASO. We didn't design this. This is all published work, right? So we just ordered up an ASO and said, what happens? So here, again, this is frataxin expression now. This is the disease gene, and you can see it's expressed in brain organoids, but it's not expressed in patient-derived brain organoids. This is treated with a control ASO. But when we treat with the ASOs that have been shown to work previously, we see super robust restoration of dystrophin expression. We also have these advanced cellular models where now we can profile neuronal activity. It turns out there's increased neuronal activity in these patient-derived brain organoids. But here's a control, and then here's the repeat targeted ASO. We see a resolution of that neuronal hyperactivity, right? So we're seeing response. It turns out these patients also have a cardiac phenotype. So here you're profiling contraction. This is kind of the typical periodicity of contraction we see in a brain organoid. And a frataxin patient, we see we see a periodicity, but it's much quicker and it's much weaker. But when we treat with the ASOs, we see, again, a resolution of this and back to normal activity. What's even more fascinating that I find about this, you couldn't maybe see it, but in the brain organoids, they have a structural phenotype to them, and the hearts kind of do too, or the cardiac organoid models. If you introduce the ASOs into the iPSC stage and then differentiate them, they never develop disease at all, or at least a disease phenotype. So you can actually show that this is the disease development process. So that's where I'm going to choose to end. I do want to highlight this work that was done here. Dr. John Means did nearly all of the ASO and organoid work that I showed you. Annabelle Martinez developed the splicing model. Tomi Passanen's our division director and the lead study investigator for genomic answers for kids. And then Paul Traynor and Toa and Lori did the fish work that I showed you. So this is we're thankful for our funding sources, and this is our mothership in Kansas City. And I'm happy to take any questions that you guys have.
We had two from the online audience. First was, if anyone's interested in enrolling, what is your recommended path? And then the second part was the two children with DMD, how did they ultimately do?
So those patients, first question, if you Google genomic answers for kids, you can find a landing page on the hospital's website, and it'll direct you through enrollment. Or you can reach out directly to me, and I can plug people into coordinators that can get enrolled in the program. You can get enrolled over the phone. It's an easy process. And then with respect to the kids with DMD, we're not treating those kids. So they are likely not receive those therapeutics. So we can do that preclinical component of saying, okay, this is hopefully going to work. But there's a challenging part with toxicity, right? And you have to show that these can be tolerated. So that's a component that's hard. The regulatory burden is incredibly challenging. Where you're seeing this be successful is in patients that are terminal late-stage disease, and you have compassionate use. When you're talking about Duchenne muscular dystrophy, and not alone in that disease, you're looking at children that have progressively degenerative disorders. And you're talking about an experimental treatment for a child who doesn't need it right now. They will succumb to the disease, right? But getting over that regulatory hurdle is just uncharted territory. And there's groups that are focused specifically on the regulatory component of personalized ASO therapeutics, and they're doing their thing and amazing work. And then the last bit of that is the cost, right? So it's, depending on who you ask, $5 to maybe $10 million to develop, GMP produce these therapeutics for a lifetime treatment. And the question is, who pays for that? And kind of the idea that we had that we can contribute to that if we can build a scalable, platform to identify patients that would benefit from this. Maybe there's some economy of scale that drops that price down. I don't know exactly how that would work, but just basically having more demand for it that maybe could contribute to that. But those are huge hurdles that we haven't overcome yet.
It seems like from everything you're showing, which is wonderful, amazing, fantastic work. Of course, people, when things work and they're so successful, people want more.
Yeah.
What, if any, is the relationship between your data and heritability of these? So when you show, it seems that you could find genetic variants, whether the parents were carriers of that or whether they were de novo patients of one kind or another. You can see them either way. Especially when you show things like ADHD, where it's not clear that how directly heritable that is. Surely there's a component of that there might be a lot that isn't. My question is, do you end up finding, wow, this wasn't carried in the parents? Or do you end up finding, most of our variants are?
Yeah, so it's a good question, and I don't have a systematic way or answer for you. A lot of the variants we focused on are de novo, because so when I say rare disease, we're talking about children that are impacted in nearly every organ system of their body. This is serious stuff. And a lot of times these kids have very, seemingly unaffected healthy parents, right? So we hypothesize that it's going to be de novo variation. So I think we get more bang for our buck in learning using that way. And there's an added challenge there about getting data from parents. So we have electronic medical records from patients, and actually accessing that data is far more challenging. We have isolated projects where we do have parent involvement in terms of cell models. And we do, one is a cardiac arrhythmia where we see a father's marginally impacted and the child has had an episode. We do see some of those phenotypes. And then I guess from my own personal experience, we have, so my child was born with a congenital heart abnormality that required surgical intervention to correct, and he's great. But we were interested in understanding he had a condition called transposition of the great arteries. So his arteries were transposed, right? So you have a closed system. This is not relevant, but like to the heart and lungs, and another closed system to the body and the heart. So you can't live that way. So they do a surgical intervention. So this form's the same in everybody that has it, but there's no known genetic effect. I didn't have it, obviously, because I wouldn't be alive if I did. But we generated cell models and did extensive single cell profiling. And you can see kind of weird ways in these U-maps of how things travel similarly and different from impacted patients versus not. So there's really cool things to dig into, but I think that part is so overwhelming that it's going to involve dedicated research. We're talking about scalable technology platforms is a little bit different than getting into those types of very deep questions.
In the precision medicine space, you showed how different patients have different responses to the drugs. So do you have to do this for every patient? Or if you know the genetic mutation, do you predict that patients with that genetic mutation will all respond similarly? Or is there other factors?
So, I can't speak directly to like what the clinician's going to be doing. I know there's certain subtypes of disease where certain drugs are going to be favored more than others. But again, a lot of our patients are undiagnosed, so we don't even know what's leading to the disease. I think, and then there's an added question, a challenge that we can't resolve right now, and that's that our process takes about 8 weeks. When a child's coming in with seizures, you can't be like, hey, hold on a minute, we're going to go think about this for 8 weeks. So catching children as they come in and being able to test them and determine if we need to is a harder thing. Where we're going to be starting on for kind of the more prospective studies is on patients with intractable seizures, where we have an established patient population that have no options. Then we'll start exploring. And I think the question you're asking, I know the question you're asking, that some of that will come to light through the process. Do we identify things or patterns in what we're approaching that maybe become predictive for the future? So I think it's going to go in tandem with our efforts moving forward.
Hi, Kanishka here. I'm a postdoc at Molecular Imaging Theranostic Center. So I do radiopharmaceuticals. I don't do this. So the fact that I understood most of it, it means you're a good speaker. So thank you. Quick question. So now we saw the imaging of brain organoid, right? And it's interesting to see how creatively you can do that. I'm thinking, is there a prophylactic way of finding out or taking a biopsy and doing it, like taking a blood sample or a brain sample, I don't know, anything, so that migraine or seizures can be found out earlier?
Yeah, right. So that's a great question, and I can't say it hasn't crossed the mind, but then it's a huge challenge to just start screening everybody. And then the last thing you want to do is, if you get a false positive or something, then, so we haven't gone in that space. But I guess we just kind of stopped thinking about it too much because it's just resource-wise, it would be so intensive that we have more to do with our time that would be more beneficial. Yeah.
So I have a couple of questions. The first one is, do you have a library of small molecules that you test regularly, or do you individualize it for each clinical presentation? And a follow-up, it's a lot of those kits actually require more than one medicament, more than one medicine to control those seizures. Do you actually test out combination therapy?
Yeah, so we're early stages on this. So yeah, we have kind of a panel that we focus on that again, this is informed clinically. So I have a clinical counterpart that I work a lot with, and in research, it's easy to read a paper and say, oh, here's all the things. And then talking to a clinician, you find out, oh, it's a little bit different in terms of the practice. And so we're really focusing on molecules and things for the anti-epileptics that are more used in the clinic. And that's kind of where we are for now. We do that in combination sometimes in the dish. I think we're going to have to think really hard, and we've got some ideas about how to approach a systematic method where every patient can come through and go through a compound screen. So we're talking about for those anti-epileptics to say what's actually going to work. Now, your first part of the question was very different. I mean, it was similar but different, right? Sure. So absolutely, we can go buy off-the-shelf commercially available libraries for FDA-approved drugs or orphan drugs and screen right through this. On a patient-by-patient basis, that's challenging. We're focusing those efforts more on specific. We have a couple I didn't talk about of specific rare diseases where we have funding to explore that particular condition. Angelman is where we're the most advanced, and so when we have those, we're doing those plan to do those library screenings where maybe we can cover a rare disease subtype and have more of an impact. That's going to be too resource intensive right now for a patient by patient. Screen.
For the sake of time, I think we're going to wrap up. Dr. Younger, really appreciate the fantastic work that you're doing and for sharing it with us and the impact that you're going to have here in the state of Missouri and beyond. So for that, we would love to share this token of our appreciation.
Awesome.
That was fabricated from lumber just about 12 miles from here, and really, signifies our efforts to really develop the next generation of therapies. And so I did have one kind of crazy question I want to ask you. When do you think this might get scaled where it's more ubiquitous? And do you think that'll ever happen where we may use a platform like this to really characterize individual makeups? Because I always make the joke that we're all mutants. We all have variants that change the way that we respond to our environment, to age. I think aging is one thing that I'm interested in. I wasn't going to ask you that question, but how do you model aging when you're reprogramming cells? So lots of questions there.
So modeling aging, we'll come back to that one real quick. Like you can actually speed up the development of these organoids through the differentiation process and build more advanced models of older organoids. These organoids are, I think, fetal to 2-year-old brain development. But you can accelerate that to get older. If you want to study things like Alzheimer's or dementia, not that you can do that really easily now, but there's methods that will advance. And then coming back, there's two things. Rare disease in particular it's not a centralized problem. We will never be a lab that brings in every rare disease biospecimen and profiles it. Like the platforms like we're developing, they'll need to be in every hospital, every pediatric health system, like across the country, across the world. And so when we started doing this, from the outset, we decided that it had to be scalable. Like that was the only way it was really going to truly be impactful, and it had to be accessible and scalable. So that's been our focus on making sure that we can achieve that. And rather than being like a proof of principle, like, hey, maybe in 30 years somebody will figure this out, let's just figure it out now. I mean, the technologies, the ASO, that whole study that we just published was a study on iPSC reprogramming, which has been around for almost 20 years. ASOs have been around for over 50 years. And the organoid space, while new, is still over a decade old, right? So these things have been around forever, but where they've been lagging is the ability to scale. And so that's where the impact was, and that's exactly what we're trying to get, is building these platforms and finding ways where we can get them built everywhere.
So when do you think every hospital will have this capacity?
So yeah, it's a good question. And one of the challenges that we face, and I think it's important to note, is that the idea of scaling and building out these reprogramming platforms, organoid platforms, there's been foundations that have been at this for 15 years. No, but
I'm going to hold you to it.
I know I'm going overboard, but the challenge is this is not an automatable process.
Right.
It's a human process. You have to have skilled human hands, and so you're not going to buy a machine that's going to do it. And so the lag is going to be getting talented researchers into these different areas to build out these platforms. The human capital is what will take time. Sorry, you asked a yes/no question or a number, and I gave a long-winded answer.
Great, it's fantastic. So thanks so much.
Great.
So please join us next month for Dr. Lucas. Thank you.